Good day and welcome all. My teammates and I are a group of fourth-year pharmacy students from Ain University, and today we're going to be presenting our project which involves the identification of potential hENT1 inhibitors using a combined approach of ligand-based and receptor-based virtual screening. I'll start by introducing you to the human equilibrative nucleoside transporters, abbreviated as hENTs. They are a family of integral proteins mostly found on cell membranes. hENTs play a crucial role in transporting nucleosides and nucleobases, which are essential for DNA and RNA synthesis. Therapeutically, the isoform hENT1 is of a great interest, being a key molecular target for drugs that inhibit its transport activity. Let me first familiarize you with the concept of inhibition. Simply, inhibitors are molecules that bind to a transporter, get stuck within its core, and prevent the passage of nucleosides. The figure on your left shows the hENT1 performing its intended function, transporting adenosine molecules across the cell membrane. Comparing it to the figure on your right, you can observe that when an inhibitor blocks the hENT1, these adenosine molecules can no longer enter the cell, and instead will accumulate outside of it. Now, more adenosine molecules are available to bind to the cell surface purinergic receptors, which in turn activates intracellular signaling pathways, mediating various protective effects. First, a cardioprotective one, which is especially beneficial in patients with hypertension and arrhythmia, as well as in those who develop an ischemia-reperfusion injury following surgeries. It also provides a neuroprotective effect, which helps with the neurodegeneration associated with Huntington's disease. And more recently, it was found to have a protective effect against lung injury induced by Pseudomonas aeruginosa. The issue in regards to the current inhibitors is that they have poor pharmacological profiles as well as many side effects, rendering them of no clinical use. And considering these limitations, there is a need to develop new inhibitors. In this study, we aim to utilize our previously validated computational protocol to identify novel hENT1 inhibitors.
In our previously published research, development of a novel filtered-based pharmacophore for the identification of a human equilibrative nucleoside transporter 1 inhibitors, we developed an inhibitors pharmacophore. And what does pharmacophore basically means? A collection of the most important features that must exist in any potential inhibitor. Now, in this project, we aim to use this previously validated pharmacophore to screen a compounds library and end up with a filtered dataset to use it for docking. Now, how we carried out our project? First, and luckily, we obtained the crystal structure of the hENT1 from the protein data bank. We optimize it using a molecular modeling software. Next, the active site or the binding site of the protein was defined in order to allow the ligands to know where they are going to bind exactly. On parallel, a ligand library preparation process was initiated by obtaining a commercially available compounds from a compound library, which in our case was Temtec. This is simply because when we later end up with our potential inhibitors, we can order them easily, skipping the synthesis process. Next, this dataset was filtered based on Lipinski's rule of 5. This drug likeness filter included a molecular weight less or equal to 500 Dalton, hydrogen bond donors less or equal to 5, hydrogen bond acceptor less or equal to 10, log B, which represents the lipophilicity of the drug, less or equal to 5. Next, the dataset was further filtered using our previously validated pharmacophore. So basically, we screened these compounds through our inhibitors pharmacophore and excluded those who didn't pass it. We started the filtration process with more than 200,000 compounds and ended up with only 52,000 compounds. Next, and by this, we finally reached to our most important step in our workflow, which is docking. Docking is basically placing the ligand inside the pocket of the transporter and modeling their interactions using the computer as it is demonstrated here. Now, we docked our filtered drug-like compounds or data set into the defined hENT1 active site. We employed a three-step protocol in our docking to allow the gradual increase in docking precision. The top 30% of each docking protocol output were utilized as an input for the next docking step. For example, when the output of our first docking protocol, which is high throughput, was approximately 47,000 compounds, we only captured the top 30% of these compounds, which equals to 14,000 something, as an input for the next docking step, which is the standard precision, and so on and so forth. Once we were done with the docking, we reserved the top tautomers of the top ranked ligands. In other words, we selected the conformations with the highest docking score from each group. Again, we started the docking step with 52,000 compounds, and we ended the docking step with only 3,631 potential inhibitors.
Later on, 500 compounds were visually inspected by us. In visual inspection, we took into consideration what are the types of interactions that were performed by our compounds, whether they are ionic, hydrogen bonding, or hydrophobic interactions. Why? Because strong interactions will assure that our compound will stay for a long time inside the transporter. Also, we looked for the compounds fitting inside the transporter. Moreover, we took into consideration to pick compounds that are balanced in terms of polarity, because eventually we would like to have drugs that are pharmacologically active. We also bear in mind to pick compounds that has different scaffolds or different shapes, just to see all possible interactions. As a result of our inspection, we ended up with top 10 hits. This table presents the best potential hENT inhibitors. As you can see here, we demonstrated the compound's name, structure, and docking score. Docking score predicts how likely our compounds likes to sit inside the transporter. Negative scores indicates that high binding affinity was achieved. And we picked here two examples that I will be furtherly discussing them in the upcoming slides. First, as you can see here, our transporter has a funnel-like shape in figure 8. And our compound, which is in purple, blocks the transporter efficiently. This blockage can be furtherly examined or seen in figure B, as it shows the top view. What's special about this compound that it has a cyano group, which possesses a unique interaction. Our compound has 5 interactions, which they are subdivided into 1 ionic, 1 hydrogen bond donor, and 1 hydrogen bond acceptor, and 2 of them are hydrophobic interactions. On the other hand, this compound has a total of 6 interactions. 2 of them are ionic and 4 of them are hydrophobic. But what's interesting about this compound that it has a pyrimidine moiety. Pyrimidine ring mimics the endogenous hENT nucleosides. Therefore, we assume that our compounds will be highly accepted by our transporter. And in this video, you can examine this blockage, as our compound blocks the transporter effectively.
To sum up, following the successful validation of a protocol for the identification of new hENT1 inhibitors, which was done last year, we proceeded to apply this protocol to screen over 200,000 commercial compounds. And later this year, we will be carrying out biological in vitro assays to ensure that the 10 compounds we selected actually inhibit the hENT1. Thank you so much for listening and hope you have a nice day.